Hi Year 12. I've put together this presentation to help with your study of topic 3.4.7 in the specification. This section is all about investigating diversity, and by that I mean how can we actually observe and measure the differences between organisms, and when we've made these measurements and observations, what can we actually do with the information that we've found out. Now this section links really closely to our topic of classification that we covered in section 3.4.5, so keep that in mind when you're listening to the presentation. So let's begin. When we think about the differences between organisms and when we sort of examine diversity, traditionally we've always looked at really easily observable characteristics. So we've looked at, does it have a backbone? Does it give birth to live young? Does it have retractable claws? Those kinds of really straightforward, easy things for us to actually observe. However, things are now changing, and because of advances in science and technology, and this has been particularly probably in the last 20 or so years, we can now actually look at diversity by looking at things like actually examining the DNA-based sequence of a particular organism, or maybe looking at their mRNA-based sequence to see and look for those similarities and those differences between different organisms. We can look at the amino acid sequence that makes up certain proteins, because we know that that directly links to what their mRNA was like, what their DNA was like. So these advances have made investigating biodiversity a lot easier in a lot of ways, but actually a lot more challenging. So we're going to explore some of these techniques and see what the future holds. So before we take a look at the kind of new methods that we're using to classify, let's just think about the old system. So the old system basically involved looking at observable characteristics, i.e. what you could see when you looked at an organism. Now we know from what we've learnt before is that each observable characteristic is coded for by a gene, or possibly a number of genes, in fact more often than not a number of genes, and they then give rise to the characteristic that we can see externally on the outside. Now what we've got to remember about those characteristics is that characteristics are not only influenced by the genetic code, but they're also influenced by the environment. So if you take something like your skin tone will be determined by the level of a protein pigment called melanin, which is decided by the genes you have for melanin production. But obviously if you spend a lot of time outside in direct sunlight, you actually affect the expression of that protein, i.e. how much melanin you produce, because you produce it to protect your skin. And as a result of that, you get the observable characteristic of your skin type would be slightly different. So it's kind of a combination of the genes that you've got and the environment. The other thing that affects that observable characteristic is if we think about any given characteristic, we can have one or two alleles for that particular gene, or we can have several different alleles of that gene. So when we look at the characteristics, observing them is a great way to kind of look at differences, but there are a number of different factors that will affect those differences that we can observe. Now, there's a problem with looking at observable characteristics, and it comes from the fact that many characteristics are what we call polygenic. Okay, and polygenic means that that characteristic is coded for by more than one gene. Now, as a result of that, the characteristic itself tends to vary, what we say, continuously. So there isn't kind of discrete boundaries where you can definitely say, right, that's one version of the characteristic, that's another version, that's another version. It's almost like on a continuous scale. Now the problem with that is that it makes it really quite challenging to decide, right, where has one version of the characteristic ended and where does the next one begin? So we've got that issue. And the other thing we've got to consider is that these characteristics will also be heavily influenced by the environment. So if you take something like height, and we know height varies amongst a population continuously, in addition to that, and that variation, is not just determined by by these genes, that variation is determined also by the environment. And so it's almost kind of deciding, right, where does the genetic influence end, and where does the influence of the environment, so what nutrition you've had, what exercise you've done, all of those things, how might they have influenced it as well. So it makes just simply looking at observable features not necessarily the best way of classifying if we want kind of a definite, this is one version of the variety we've seen and this is another version. So that led scientists on to think, right, is there a better way that we can look for to classify in the future? So to make things easier, what's kind of happened and what's developed is that rather than inferring that there are differences between the DNA from the characteristics that you're observing, actually we started to directly observe these differences. Okay, so we actually look at the DNA-based sequence and see, right, how does that vary between that organism or another organism? Or we look directly at the differences in the proteins. Now we can make that link because we know that the DNA-based sequence directly determines the amino acids that are produced and hence the protein that's produced. So all these advances in DNA technology have totally revolutionised how we classify.